webinar today. So today I'd like to focus on the use of surface analytical methods for biomedical applications, as well as a focus on nanostructured materials. So first, let me introduce myself briefly. I'm a professor at Case Western Reserve University in Cleveland, Ohio. We are located at the Kent Hill Smith Building. And anytime you're in the Cleveland area, let me know, and I'll be happy to host you. When we talk about nanotechnology, nanostructure, it is indeed an interdisciplinary field. As you can see here, we deal with materials and their properties at the nanoscale. Atomic to molecular level, if you want to put it that way, ultra-structured, ultra-thin films, nanostructured, nanoparticles are some of the key words. Some of the popular materials could involve carbon-based materials, atomic layer deposition, dendrimers. But in focus, with respect to nanoscale at the biomedical, biological interface, we're dealing with proteins, DNA, peptides, membranes, and interfaces for interrogating or investigating biological and biomedical phenomena. Thus, nanotechnology is a good platform to understand a biological phenomenon. In terms of interfaces, very important for us to realize that the high surface-to-volume ratio of nanomaterials interfaces well with the phenomena of biomolecules on surfaces, membrane transport systems, design of smart bioimplant surfaces, biomedical application of polymers, tissue growth, and even surfaces involved with cell growth and chemotaxis. Our group, in fact, has been involved very much with the biomedical space, whether it be investigating DNA adsorption on surfaces, as shown here, where we use atomic force microscopy, which Park AFM Systems enables us to do a lot of our interesting work. Here we can see actually DNA that has been double helix DNA that has been adsorbed on the dendrimer surface in a multi-layer deposition pattern. Other applications that we've investigated include modification of titanium alloy surfaces and looking at sol-gel methods to improve the adhesion of fibroblast cells. Or in the case of antimicrobial surfaces, we've demonstrated that graphene oxide, or modification of pegylated linear dendrite systems and surfaces have both anti-biofouling, antibacterial, antimicrobial properties. Here, for example, it's exhibited where we look at the antimicrobial properties against E. coli of these biomedically relevant surfaces. So our group has been involved very much at the biomedical space and interrogation of surfaces, even using nanomaterials. So what you can see here is that tools are vital, important in understanding this relationship between cell biology, physiology, or biomedical problems with the use of materials, whether it be implant, alloys, surfaces, semiconductors for measurements, or even polymers for tissue engineering, or a memory-shaped devices or bioimplants, or even stents preventing, let's say, thrombosis or coagulation of blood. The interaction up to the nanoscale between biorelevant problems and materials is important, and therefore having excellent tools or method allows us to understand that relationship better. So I'd like to start by focusing on interfaces. So interfaces, you have basically a solid, liquid, solid, air, liquid, liquid, or a classification of 5 different interfaces. And within that interface, you have various phenomena, whether it's electrostatic, van der Waals, that involves attraction or repulsion. And therefore, surface chemistry, surface morphology is important for the interrogation of cell systems that adhere to functionalized surfaces, which includes, of course, your stents, your bioimplants, and other biomedical devices. So that means the cell is basically inhabiting an unfamiliar environment, whereas in the human body, it's used to encountering different types of tissues, hard tissues or hard surfaces that are biocompatible or communicate very well. So understanding the surface phenomena involves looking at the first principle. So this means that the distance of that interaction, which can be quantified by methods like surface force interaction measurements or atomic force microscopy methods or different welding phenomena, means that the bacterium interacts with the surface as a function of distance. That distance, as it goes closer, becomes attractive to repulsive. And that means surface functional groups like hydrophobic groups, electrostatic interactions, or specific biointeractions are important for making a cell compatible. Of course, one of the easiest tests this can be done is by using proteins as a way of understanding fouling or non-biofouling properties. So if you look at this typical interaction between a cell and a surface, certain types of proteins, for example, RGD peptides, this is a particular sequence of amino acid that promotes extracellular matrix adhesion and in general a cascade of events that enable cell adhesion and finally cell growth. In fact, a surface has several stages of response for a cell in that to interrogate the surface being friendly for the cell, sometimes a type of biomolecule adsorption takes place. Then the cell deposits or interacts on the surface in chemotaxis type of manner, meaning the chemical cues enable the cell to first adhere and feel comfortable on the surface. And then over time, that cell has a multiplier effect that results in tissue regeneration or integration on the surface. And as you can see here, both the surface chemistry as well as actually the morphology or the roughness of the surface and its thermomechanical properties in many studies play an important role. And this can take from minutes to days. Now, a bioimplant is basically a foreign material that has a certain degree of compatibility, whether it's mechanical or electrochemical, that is corrosion, or in terms of its ability to allow the cell to grow on the surface, as the previous slide shows. And this much has to do with the interaction of the cell and the proteins and the glycoproteins present to interact with the different chemistry, morphology, crystallinity, or texture of a particular implant. Understanding so prevents fouling or rejection of the foreign body or implant by the individuation, or in the case of design of better bioimplant devices, makes for superior and long-term adhesion or properties of the bioimplant. Let's say if you have an artificial hip or a bone replacement that you want to last for 10 to 20 years, understanding all these phenomena is important. Cells do move on the surface, and integration and understanding this chemical gradient term chemotaxis means that you want to understand how the cell selectively absorbs and migrates or forms different shapes at the interface. And there's much more involved to that. but we will not have time to go over all those elements. On the other hand, the surface or foreign body can trigger an immune response that can be synergistic or non-synergistic. So foreign bodies, proteins, viruses, bacteria typically can have a humoral or cell-mediated response, which can eventually lead to rejection. Okay, and the presence of different antibodies and macrophages, as in the case of, let's say, wound healing or infection, speaks to that type of understanding. So, for example, in inflammation, there are several stages that can involve white blood cells, the generation of macrophages, and encapsulation of the foreign body. Probably a manifestation of this that you're more familiar with is pus, which is that fluid generated whenever you have an infection that needs to be removed in order for the body to respond better towards healing, or it's actually a stage of healing. So anyway, with that introduction, you can see why biological information is quite interesting when you match it with the materials world. So let's first take a look at the membrane system. So a membrane is a lipid bilayer composed of lipids, protein, membrane proteins, glycoproteins. This has a very important function in terms of self-transport of ions, as well as its ability to interact with other cells, eventually forming a tissue. So one can say that the lipid bilayer is an active membrane that allows transport or rejection of certain species, molecules, biomolecules, and therefore understanding the transport properties in an artificial manner enables us to quantify this response. So many tools have been generated to look at solid support biomembranes that enable the explicit study of these transmembrane proteins and its function towards ion transport, or in passive and active transport of other biomolecular species. That means the membrane has to interact, let's say, with a surface that will enable you to measure, let's say, electrochemical properties or ion-gradient properties. So the way that's been done is basically to take an artificial black lipid membrane that's been reconstituted and then provide an interface between that hard surface, let's say with the use of hydrated polymer or functionally lipid tethered systems that maintains the integrity of the membrane against a hard surface. So membrane is a very lipid bilayer is a very dynamic system, and therefore trying to preserve its viscosity, viscoelastic nature, etc., is important to actually measuring closer to in vivo interactions. As you can see here, there's a lot of lateral diffusion within these artificial membranes, and therefore making it liquid-like or liquid expanded-like. Enables you to preserve the property of a membrane. Many types of work has been done, and this is actually older literature, where one can corral these membranes artificially and investigate diffusion laterally through the use of dyes. And that means that the membrane can be patterned and investigated individually or used in an array system as shown here, that enable better labeling experiments. However, membranes can be also integrated with a semiconductor device. The semiconductor device enables, for example, techniques like electrochemical impedance spectroscopy or other types of electrical chemical methods, or measuring field effect transistors and elicit the response of cells. In particular, neural cells, or even different types of barrier functions within the lipid bilayer measured as a gradient on the conductivity, ion and electrical conductivity between the substrate and the membrane. So that shows you that this type of measurements are very sensitive to that surface interaction. And in this case, as shown in this image, this can be done by providing a polymer insulator, quote unquote, which provides a soft landing for the membrane against the hard surface. Another technique that's very important is nanomanipulation. This can be done basically by using tools to poke, to measure, to surgically, quote unquote, reconstruct. A lot of these nanomanipulation tools, although this is not my expertise, is very important because it allows you to measure and manipulate living cells as observed in a microscope. So a micromanipulator, nanomanipulator are essential tools. So again, this section just summarizes the importance of the interface and the use of tools to interrogate that method. Now, looking at more macroscopic type of behavior in both in vitro and in vivo methods, let's classify this as spectroscopic and microscopic in nature. This means that you can apply the spectroscopic methods either in vitro or in vivo studies. Of course, it's desirable to do more in vivo studies, but in vitro studies enable us to access better tools whilst trying to keep the ex situ or external environment closer to in vitro studies. In vitro studies require tools that probe a living system, and that means that you need to pay attention to the minimally invasive methods that can preserve the health of the living system, whether it's an animal model or even a human body. Another way of classifying the studies is whether we're dealing with in situ or ex situ. In situ means in real time, invasive. Ex situ meaning after the fact, monitoring or after the event has occurred. And so understanding the nature of the studies, whether it's in vitro, in vivo, ex situ, in situ, allows us simply to classify all these methods. So let's first try to understand living organisms. Let's start with proteins, viruses, bacteriophages, and cells. So if you're not familiar with these terms, or we can also divide this as eukaryotic or prokaryotic systems. Essentially, in terms of scale, the smallest pseudo-living systems that we encounter are viruses. These are basically assemblies of proteins in a symmetrical manner, as shown here. Sometimes they are called bacteriophages, which are larger than viruses. Viruses can come from plant or animal sources. And then we have the living cells. Within the living cells, we have organelles. These organelles, including the mitochondria, chloroplasts, lysozyme. The corresponding dimension, as shown here, is in terms of submicron to several microns. The micron sizes here, as shown in this table, differentiates organelles and living cells. Living cells are typically between 8 to 15 microns. Bigger cells or parts or tissues can range from several hundred microns as well. On the other hand, I forgot to mention, most viruses and bacteriophages are in the order of 20 to 40 nanometers. Now, we are dealing chemically with these living systems basically as organic chemistry. So you have the chemistry of carbon, hydrogen, nitrogen, oxygen, sulfur. We classify those biomolecules as amino acids, nucleotides. Amino acids build up polypeptides or proteins. Nucleotides, nucleobases build up DNA or RNA. And then, of course, viruses are units that are based on the assembly of proteins with nucleic material, RNA or DNA, that are part of the complex or as carriers of this genetic material. So here, as you can see, again, for comparison of size, for those particular biomolecules that I mentioned. So having said that, you see why spectroscopy is important. Spectroscopy allows us to distinguish the presence of amino acids, lipids, proteins, carbohydrates, fatty acids, simply by the functional group that one can identify with IR. In fact, IR together with Raman, as shown here, are quite complementary in that spectroscopic imaging, spectroscopic analysis are often used to characterize biomolecules present in cells as well as in fluids excreted from living organism. So take this simple IR spectroscopy utilized in nephrology. In nephrology, which is involves the study of the urinary tract, urine. Here you can see IR spectroscopically easily used to classify the presence of urea, creatinine, uric acid, glucose, albumin in urine. Okay. On the other hand, with Raman, you can use Raman spectroscopy combined actually with Raman imaging to look at the presence of Raman-sensitive functional groups, and those are typically phenyl rings, double bonds, or a particular frequency that are not readily observed by IR, as you can see in the range by which Raman is very sensitive compared to IR. Okay, and of course, X-rays are used very much for structural biology. So from spectroscopy, let's go to imaging. So in imaging, obviously, we can talk about scanning electron microscopy. We'll have examples of atomic force microscopy, but let's start with simple optical microscopy. So in simple optical microscopy, which is very much used in pathology, you simply look at the histology of different types of cells or bacteria that can be used to classify eukaryotic and prokaryotic cells or diseases. So when you combine imaging with spectroscopy, you can have instruments like this FTIR imaging setup. Now, FTIR imaging is possible simply by using an optical microscope and an IR detector or IR setup to do single-point spectroscopy. However, in an imaging setup, one can use what we call a focal plane array detector, which, to make a long story short, this technology is similar to what is found in military technology called the Stingray missile. It's essentially an IR detector that captures the spectra over a wide area of a pixelated detector. Okay, so with that definition, one can use it for mapping or doing IR mapping of an optical image. And this enables it to be very useful for chemometrics, tissue and disease detection, spectral information from histopathology samples, etc. And I'll show you some examples. For this case, you have a cell, tumor cell that has been removed from a chorioalveolentoic membrane. So you can see here the sequence of the tissue. After the tissue has been removed, you have this histology sample observed by a normal optical microscope. And then you zoom into that image, which enables you to do the FTIR imaging setup. So the FTIR imaging setup as shown here is essentially a false color map of the different pixels, and I'm not sure how many pixels were used here, showing that in this region you have this particular IR signature. Now, the sequence of boxes here essentially is the distance that was taken to gather each of these spectra in the mid-IR range. And in this case, I think it's sampling for the presence of proteins as well as different types of polysaccharides or glycoprotein interaction that can be diagnosed or analyzed from this spectra. However, even though this spectra is shown clearly in this two-dimensional diagram, this IR map shows the distribution of those species within a space. Now, in chemometrics, this can be done by doing time sequence measurements, which allow you to gather the spectra in one dimension, and then on another dimension, look at the changes in the distribution over space. So to make a long story short, IR imaging is very useful for further analysis in histology, in that with normal optical microscope, you only have a static experiment on the cell or tissue. However, with chemometrics and IR imaging, you're able to do time or frequency dependent measurements. Okay? And therefore, one can develop different types of algorithm with this data that can be gathered over time. One can use cause-follower mapping to investigate the distribution. And we usually do this by fixing the frequency over a particular range and then looking at that specific frequency on the image. Here is a lymph node tissue specimen that was sampled in a 3 by 10 millimeter sample, which is measured in microns. And then here you can see here the distribution for very specific frequencies that were assigned during the chemometrics analysis. Okay, so let me skip these several examples on IR imaging so I can go to other types of methods. So what you can see here is basically IR imaging is very important for histology and pathology, in that you can extend it towards spectroscopic mapping and quantitative analysis. Okay, so again, let me skip this last few slides. Now let me go to the use of quantum dots and fluorescence microscopy. Now, other than the use of dyes for fluorescence microscopy, two-photon spectroscopic methods, fluorescence methods, excitation methods in fluorescent microscopy, quantum dots play a very important role in that they are basically fluorescent materials that are very stable against photobleaching. Moreover, as you can see here, quantum dots, for example, things like cadmium selenide or zinc selenide, give different emission colors by virtue of their size. So as you can see here, by varying the size of the nanoparticle or quantum dot, you get different emissions. Furthermore, the surface itself can be functionalized or bioconjugated to have some specific biomarker application. Here, again, it shows you some of the absorption and fluorescence characteristics, and the fact that these nanoparticles are in the order of several nanometers means that they are in the region closer to that of enzymes or particular biomolecule assemblies. So what I'm trying to point here is that quantum dots are very useful as alternative agents for the use of organic dyes. So let me skip that and show how two-photon excitation fluorescence microscopy works. Basically, this experimental design allows you to use a single excitation to make visible several dyes within a setup. And again, because of lack of time, I cannot explain the mechanics of this further, except that in this particular image, you can see a endothelial cells that have been labeled on different parts of the cell, from the cytoplasm to the nucleus to the actin and mitochondria, so and then intercellular matrix. Different dyes were integrated or absorbed to these different parts, and the use of two-photon excitation enables you to see all these dyes in one experiment versus one dye excited with one type of light. So two-photon experiment absolutely allows you to get these beautiful images with lots of information. Now, I mentioned about bioconjugation. Bioconjugation is simply attaching proteins, peptides, biomolecules on surfaces of, let's say, these quantum dots through a number of chemical attachment methods, and that makes them very useful for labeling, or even what we call theranostic experiments. That is a combination of therapy and diagnostics. And the surface of the quantum dot, depending on the bioconjugation, can be made specific or non-specific. So just to summarize in this busy view graph, some of the bioconjugation techniques that have been used to functionalize quantum dots. Quantum dots can have different types of ligands, whether it's phosphonate, thiol, amine, alcohol. So that provides a way to attach these biomolecules on the surface and functionalize it. So as you can see here, one example is you can functionalize DNA on a quantum dot using a thiol chemistry. Or one can functionalize oligomeric phosphines directly on the surface through the phosphorus group, okay, and so on. So there are many types of bioconjugation methods to make those quantum dots more biospecific. Okay, let me skip that. So the reason is that if you do this, the quantum dots can be very specific for the part of the cell that you're trying to image. So here you can see different colors: cyan, magenta, green, blue, which is used to label the nucleus, the different types of proteins, the mitochondria, microtubules, and the actin filaments present. And this was all done in a two-photon imaging setup, as I mentioned earlier. Now, quantum dots are also useful in that one can label them with growth factor receptors, and they can be useful for specific growth or biodistribution of a probe on a cell, or incorporating this, let's say, in a mice cell model. That is, this type of labeling experiments can enhance the contrast versus an unlabeled sample for a specific biodistribution within a animal model. Or specifically here, you can see the enhancement of adding a growth factor on a quantum dot versus a quantum dot that has not been bioconjugated. And here, just for a general comparison, you can see the enhancement in the imaging setup. Now, for lack of time, let me skip some of these other applications of the quantum dot. Okay. And apologize for that. Okay. In fact, this is an interesting setup, and this is my last slide for this section, which shows that a quantum dot properly labeled with gadolinium chelating groups can be useful not only for a two-photon fluorescence microscopy setup, but also useful for MRI imaging. Especially the gadolinium complex provides the contrast that is very useful for magnetic resonance imaging. So in this case, the same quantum dot that has been integrated can be visualized in two types of microscopy setup. So with our remaining time, what I'd like to focus is simply give you an overview of the things we have been doing in our group. So for example, we've been interested in the adsorption of DNA, massive adsorption and high density DNA on thin film constructs that may be useful for DNA non-viral drug delivery, or simply for using it as a platform for investigating DNA hybridization or DNA packaging. One of the key materials we use here are basically polycations that adsorb on DNA, which is a negative, considered a negative polymer. Polycations have been used for a long time to stabilize DNA, their aggregation properties. And in this case, we use what we call a layer-by-layer technique. So I'll just show how we did this. The layer-by-layer technique is basically alternating between a polycation and DNA, and we can incorporate dyes, and then monitoring it with a number of techniques. So for example, we use a dendrimer or PDADMAC to make layers of DNA derived from sperm whale. We monitored it using surface plasmon resonance spectroscopy. The layer-by-layer adsorption can be seen as a shift in the reflectivity angular scan. So here, from 5, 10, 15, 20 layers, we can measure the exact thin film thickness and refractive index. In this case, each construct is about 4 nanometer in thickness with a refractive index of 1.5. So this was done quantitatively using the SPR method. However, we can visualize directly the adsorption of DNA in layers. So in this case, the topmost layer was successfully investigated by AFM, atomic force microscopy. You can see here that if we have the last layer being that of the adsorbing polycation, you see various domains. On the other hand, if the last layer adsorbed was DNA, you can clearly see the adsorption of DNA double helix on a surface. In fact, this is consistent with a random walk of a relatively rigid polymer on a surface, which scales with the molecular weight. So the higher the molecular weight, we can see what we call a change in the radius of gyration. And statistically, this corresponds to the DNA that we actually use, which is about 50 base pairs. However, when we use a different polymer, what we call a dendrimer, we did not observe this type of a random walk, in that the dendrimer basically disorders or incorporates the DNA in this globular structure. This globular structure is basically the very strong cationic interaction of a highly compact and charged dendrimer versus a polylysine. So again, we monitored SPR. In this case, we also use a UV spectroscopy to monitor the additive effect of the number of layers. So from 5, 10, 15, we see an increase in absorption. And then to verify the presence of the DNA on the material itself, we turned to X-ray photoelectron spectroscopy. X-ray photoelectron spectroscopy is a sort of elemental analysis on the surface. And to confirm that we indeed have DNA, we have a good peak of the phosphate group or phosphorus element with a binding energy of about 130, growing with the increasing number of layers. Again, an important information we can derive from FTIR imaging. This is FTIR imaging of a 64 by 64 array. So what you can imagine is we take this optical image and then use the IR imaging setup, which shows the distribution of DNA corresponding to the amine group at 3400 on the surface. So 3400, this shows how the DNA is distributed with increasing intensity as you go to more orange color. 1650 corresponds to the amide group, so we can see the distribution of the amide group, and in another area corresponding to the amine group. So what you can see here is a non-uniform distribution of the DNA versus that of the amine group. And there's no specific correspondence with the um, uh, uh, cation and anion complexation between the PAMAM and the DNA. However, we can use also AFM again to investigate the use of different polymers and the ability of DNA to absorb on the surface. This is shown here. If we use different types of polymers, a diablock, a triblock, a polydadmac, pdadmac, homopolymer, you can clearly see the influence of the polycation in causing disorder or different morphologies on the DNA. Basically, from this image, you can see that the pdadmac polycation is very good in preserving some of the random walk observed with that of the hybridized DNA. So let me turn to the next project we did with layer-by-layer sol-gel modification of a titanium oxide bio-implant. We did this using what we call a surface sol-gel layering technique, where we modified the substrate with a monolayer or thin layer of a titanium alkoxide sol-gel layer, which we can then build up into multi-layers as shown here. So from 1 layer, 2 layer, 3 layers, we can easily build up that thickness. And as shown here by AFM and SEM imaging, we build up these layers with a particular morphology. Now what we can do with the hydroxyl groups, or the last layer, is basically to do what we call a self-assembled monolayer technique, which involves that of a functionalization with a silane agent on the surface. And this can be done with an amine or an octadecyl or a perfluorinated system or a benzophenone derivative. We did this actually in order to modify the surface wetting properties and the ability of the surface to prevent protein and fibroblast cell growth. Again, AFM is a vital tool that allows us to look at the morphology, compare that with the roughness and the wetting, and then calculate the surface coverage. And then finally, because of this controlled chemistry at the surface, we are able to use sol-gel layering in order to control the growth of cells or non-biofouling properties of cells on the surface. So what you see here are basically titanium alloys coated with gel, producing that rough surface. And then by incubating it with fibroblast cells, we can see how the fibroblast cells grow on the surface on the first hour and then after 24 hours. So the conclusion of this work or paper is that we are able to modify surfaces and utilizing techniques like AFM, SEM, fluorescence microscopy to investigate that successful surface modification and cell growth. On another project, we were interested with the use of polymers, in particular polyethylene glycol, in producing what we call non-biofouling surfaces. So pegylation is a well-known technique for preventing absorption of proteins on surfaces, basically reconstructing surfaces that are more friendly to water than the biomolecule. Therefore, the solvation or the ability to retain water is much higher than that of the enthalpy for absorption. There are many studies using different types of polymers and self-assembled monolayers as shown here. We had focused on a family of polymers called linear dendron systems, where we can vary the distance between these pegylated linear chains by controlling the size of the anchoring group. And as shown here, the bigger the anchoring group, the higher the surface. So synthetically, we made this carbazole-containing dendrons with a PEG or a TEG oligomer group, and then deposited them on the surface by electropolymerization. So in principle, we can have varied distances between this TEG depending on this anchoring group. And what we have found, again, AFM was very useful here to look at the extent of protein rejection and absorption, and in congruent with a technique called quartz crystal microbalance, we were able to quantify the amount of protein absorbed. So you can clearly see here that the electropolymerized PEG dendrons did a very good job in rejecting the protein, whereas a controlled experiment of a mercapto, butane, or even bare gold shows more protein absorption. Another way we quantify this is by the use of surface plasmon resonance spectroscopy, monitoring the adsorption kinetics as shown here, where again we found that the control of the distance between those PEG on the surface resulted in lower adsorption profiles as compared to the control. Now, in comparison, we can actually use ex situ methods like fluorescence and AFM or XPS. So we also developed this type of polymers which can be functionalized with this polymer pegylated brush component on the surface. The idea is to use the surfaces to reject proteins as well as cells, fibroblast cells. And indeed, or again, we were successful in doing this. And this was done using XPS, fluorescence spectroscopy, and again AFM to corroborate the rejection of high protein resistance by using those pegmetaculate brush-derived surfaces. So I think we have our PowerPoint presentation simply crashed. Let me see if we can pull this up again. I think this is the first time that this has happened in this webinar series since we started it. Let's see if we can.
Yeah, well, I guess there's a first time for everything. Well, while the professor tries to get the slides back onto the screen, I'd like to remind everyone here that if they came in late or if they'd like to refer the presentation that Professor Advincula has done today or any of the previous sessions that we've done with the professor, they're all going to be available at parcafm.com. We've archived them onto YouTube, so that way you can stream them at a later time if you ever need to go back to refer to something. So it takes about a week or so for our counterparts in Korea to get it edited, to go through post, as it were. So after about a week after today's airtime, it'll eventually be on parcafm.com. You can see it in the Learning Center and Nano Academy sections on that website. If you have any questions at all about how to find it, it is a little buried on our website. You can always email me. Again, my name is Gerald, and I can give you a direct link to those sessions. I'll go ahead and leave my address in the chat section. Other than that, after today's session, there will also be a follow-up email for feedback. So if you have time, we'd greatly appreciate you to see if you have time to complete the feedback form and give us some of your thoughts about how the sessions have been and what you'd like to see moving forward. It looks like that the slides are now back up on the screen, so let me turn things back over to Professor Advincula.
Yes, thank you, Gerald. So actually we're almost done. I'm just going to show you one case study or one project remaining. So anyway, these methods we use to, in particular, look at the or quantify the amount of protein rejected or protein resistance of these films. So techniques like X-ray photoelectron spectroscopy or XPS allows us to quantify the amount of proteins present based on the binding energy between carbon and nitrogen. AFM, as you can see here, the difference between a surface that has been absorbed with protein versus none, or looking at the fluorescence image. As you can see here, the control with bare gold or just the chain transfer agent shows lots of protein that's been labeled with a green dye. They have absorbed, whereas the PEG film, there were barely any. So this bar chart shows those quantification. On the cells adhesion studies, here you can see bare gold, the chain transfer agent, and the polymer brush grafted, and the fibroblast cells as investigated by SEM. So you can clearly see here that on the PEPEG MEMA-derived surface, very little or if none of the cells were absorbed. So the last topic that I actually wanted to cover, and I'm done with this last topic, was molecular imprinting. So in molecular imprinting, we utilize techniques like cross-linking, polymerization, electropolymerization to mimic enzymes or receptors, where enzymes and receptors are very specific for a molecule that binds to the surface cavity, as shown here, and then it elicits a response or a catalytic effect. So the way we do with molecular imprinting is basically we know the analyte, and then we build a artificial enzyme or artificial receptor around it by polymerization. So it's like a lock and key mechanism where we actually have the key but we don't have a lock. So we need to produce a lock. On the other hand, in a normal enzyme, you essentially have the lock and the key is the substrate. So in this case, by fixing the material on a surface, we can do sensing experiments, in this case by surface plasmon resonance spectroscopy. So when the analyte binds on the cavity, that binding event can be detected by this transduction mechanism as an optical signal. So just to show you the materials we utilize these monomers or electropolymerizable monomers that can then be complex with the analyte that we are investigating. In this case, we investigated drugs, different types of analogs of caffeine, and then we utilize the molecular imprinting. The result, as shown here, is that the original template, whether it's naproxen, theophylline, or paracetamol, has very high binding coefficients or binding events as compared to the non-conforming or non-specific analytes that were introduced. So you can see how powerful molecular imprinting is in that you can make very, very specific interaction on surfaces. And in fact, this has resulted to many publications in my group and different methods of sensing as well as separation. So in fact, believe it or not, this is my last slide, and I'll be happy to answer any questions you may have. Okay, and feel free to email me, contact me regarding today's session. So actually there are a number of things that I did not have a chance to talk to, but biomedical field relies a lot on very good analytical tools to understand the biological phenomena and the interface of biological phenomena with materials chemistry. So here at CASE, we collaborate with a number of people in the medical side of the campus, as well as other collaborators utilizing tools like this. Now, in the other coming webinars, we'll be focusing on different topics. Again, anything you'd like to ask or even know more about the coming topics, feel free to communicate with me. Certainly, atomic force microscopy is a very important tool, and we utilize it, and we are utilizing it in almost all of the projects that we do in our lab.
Certainly. And again, the professor's email address is there on the screen. If you're not able to read it clearly, it's rca41@case.edu. If you have any questions about the atomic force microscopy, the AFM systems that Park Systems provides, feel free to visit our website, again, parkafm.com, or you can send me, Gerald, an email, and I can definitely connect you to the right people to answer your questions. So we are just about out of time for this hour. Looks like no questions have come in, but again, those are our email addresses if anything should arise post-session. And I believe that will draw our time to a close here. So on behalf of everyone here at Park Systems, thank you very much for attending today's session, and as well as On behalf of Professor Advincula, thank you very much again, and we hope to see you again about this time next month. Any closing words, Professor?
Well, thank you, and have a good day. Have a good evening.
All right. Thank you very much, everyone. We'll see you next time.